Alright, so we're going to go ahead and talk about the four organic macromolecules. We'll just cover carbs in this video. But if you remember that, let me grab my little marker here, that organic, I'm going to write this over here, organic, my writing on here is going to be a little funky, but you guys should be able to follow along. Organic, I'm just going to abbreviate macromolecules. Have carbon-carbon and carbon-hydrogen bonds. That's what makes them organic. That's the definition of an organic molecule. In carbohydrates in particular, they're going to have carbon atoms, hydrogen atoms, and oxygen atoms in a very specific ratio, in a 1 to 2 to 1 ratio. And we're going to see that as we look through some of these examples down here. Let's start with let's look at the glucose first. So monomers are single units. Again, monomer means one, mono means one, and saccharide means sugar. So the most fundamental kind of building block of your carbohydrates are your sugars. And if you kind of look on the back of any food label at the carbs, you will see a little section devoted to sugars. And that's what they're talking about. They're talking about these monosaccharides and some of these other saccharides that we're gonna talk about today. One of probably the more common ones that you guys are familiar with is glucose. So if you go ahead and take a look at this glucose molecule and you just literally go through and you count all of these atoms down here, you are going to find a C1H2O1 ratio. And they've numbered the carbons for you, so there are 6 of them. And if you go through and you count the hydrogens, 1, 2, 3, 4, all the way around, you'll actually find that there are 12. And if you count the oxygens, 1, 2, 3, 4, 5, 6, there you go. So if you have a molecule that's unlabeled and you go ahead and you count atoms, it's kind of tedious, but if you did it and you found C6H12O6, you'd say, hey, that's a carbohydrate, that's actually going to be glucose. And there's actually another example of C6H12O6 being not glucose, and we'll get there in a second. But you're going to notice that this is a straight chain. This glucose molecule is a straight chain. Straight chain glucose is usually what you have in a dry environment, just like powdered glucose. Let's see, you're also going to notice, if you look at this glucose molecule, that attached to each of these carbons are either another carbon molecule, oxygen, or hydrogen. And since we just talked about water, I want you guys to pay close attention to these OH groups, right? These are your hydroxyl groups. These are hydroxyl groups. Every time you see a hydroxyl group, you should immediately associate that molecule with polarity. And here's why, right? When you have an oxygen attached to a hydrogen, which is what a hydroxyl group is, right? Does the oxygen and the hydrogen share electrons equally? No, absolutely not. The oxygen is going to be much more electronegative. It's going to hog those electrons. So the overall charge here on the oxygen is going to be slightly negative, and the overall charge here on the hydrogen is going to be slightly positive. And what that's going to do is it's going to attract other polar molecules, just like water. So water is going to want to form these hydrogen bonds between the oxygen of the water and the hydrogen of this hydroxyl group. And every time you just kind of drop glucose or these any compound with a hydroxyl group on it, you just drop it in water, those water molecules are going to find these hydroxyl groups and they're going to kind of surround them, oxygen first, because oxygen's electronegative, and it says, oh, hey hydrogen, you got this nice positive charge on you? I am a nice negative oxygen. I'm going to come right up next to you. So that's what's going to happen every single time you drop these things in water, right? So you should immediately be able to tell if a molecule is polar, right? So a glucose molecule is a polar molecule because it has those hydroxyl groups. If and when you do that, it doesn't actually stay in this kind of straight chain. So as soon as you drop things like glucose into water, they actually are going to fold up on themselves. So this second picture is glucose in solution, in water, in solution. And we care about this because we're doing biology here. And your cells are mostly made of water. They're 72+% water. So any kind of monosaccharide in your cells is going to be found in this kind of hexagonal kind of formation here. And you'll see everything is exactly the same. You've got carbon 1, carbon 2, carbon 3. All 6 carbons are still here. The 6th carbon is actually kind of outside this ring. There's an oxygen in the middle of it. It's part of the ring. But this is the in solution. This is in your cells. This is in water. Right. All right, so let's go ahead and scroll down a little bit. Do this. So yes, glucose is C6H12O6, but you're also going to have different monosaccharides having the same formula. They're also all going to be C6H12O6. And if you, actually, let's go ahead and complete this. Galactose kind of got cut off here. We're going to draw, I'm going to have you guys draw this in. There's a hydroxyl group right here. Let's add that. And there's a hydrogen right there. But if you count all the atoms in each of these monosaccharides, what you're going to find is that there are 6 carbons, 12 hydrogens, and 6 oxygens. They all have the same chemical formula. So what's the difference? If they all are C6H12O6, what differentiates these from each other? And the answer is just how the hydroxyl and the hydrogen groups are situated. So if we take a look at these first two, if we look at galactose and glucose, what you're going to notice is that on carbon number 4, this 4th carbon right here, you have the OH group kind of on top and the hydrogen on the bottom. It's the opposite for glucose. You have the hydroxyl group down here and the hydrogen up here, so they're flipped. And that is the only difference, and that's enough of a difference to make them different monosaccharides. And they have different, slightly different chemical properties. Anytime you have galactose and glucose attached to each other, we are going to call this lactose, and we'll talk more about lactose and disaccharides in a minute. That's lactose. If you take a look at fructose, again, there's C6H12O6, but it's more different, right? Here you have kind of this hexagon shape with glucose and galactose, but here you have a pentagon shape. But if you count all the atoms, you're still going to have C6H12O6, right? So it's still the same formula, it's just the arrangement of the atoms that differentiates these single, or mono is single, and saccharide means sugar, these single sugars. So again, this is like one sugar, one sugar, one sugar. Right? Just for completeness, let's take glucose and fructose. If those two are bonded together, that's actually called sucrose, and that's table sugar. That's the same kind of sugar that we put in desserts and we make them all sweet. It's when you think of, sugar I'm going to consume, you're probably thinking of sucrose. All right. And again, these are disaccharides, which we're going to discuss now. All right, di means 2, and saccharide means sugar. These are 2 sugars. All right, so let's go ahead and just see what happens when we add these sugars together. So I'm going to go ahead and scroll back up. So we're going to take glucose and we're going to take fructose. Again, these are 2 monosaccharides, and we're going to link them together. So how are we going to do that? How are we going to do that? We're going to do something, or we're not, enzymes are, called dehydration synthesis. And another word for that is a condensation. I'm going to write it up here. A condensation reaction. These are synonyms. They mean exactly the same thing, so don't get freaked out if you see either one. They're identical. They're synonyms. Again, to dehydrate something, again, you can probably guess, is to remove water. And to synthesize something is to bring together. So in order to combine these two monosaccharides, we're going to remove a water. And here's where this water molecule comes from. You have the hydroxyl group of carbon number 1 of the glucose and the hydrogen from this carbon on fructose. That's actually where the water comes from. If you remove that water molecule, they're going to form a bond, and I'm going to show you where that bond is formed. All right, so we're going to say form bond. And note that an enzyme has to do this. There are enzymes in your body that specialize for specific reactions. There's a specific enzyme that facilitates this particular reaction, right? So we're removing the water, and if you look at the final product, again, this is sucrose, it's a disaccharide. You'll see that this oxygen right here, let me just change color. Oop, that changed everything. No, that's okay. That's okay. We'll just stick with blue. So this oxygen right here attaches to this carbon. You form a bond right there. That's a new covalent bond. That's the new bond that forms, right? Because water has left. So you formed a new covalent bond, you've linked glucose and fructose together, you formed a disaccharide, in this case sucrose. The only additional thing I want to add here is that this particular covalent bond, this new bond that formed, has a specific name. And the name of that bond I'm going to write over here is called a glycosidic linkage. And there are different types. There's like 1, 4, alpha, beta. You don't have to know any of that kind of stuff. But it's a whole variety of bonds called a glycosidic linkage. One thing I do want to point out now is if you're looking at these diagrams and you're like, well, where did all these extra things go? Because if we scroll down back to our macromolecules, or sorry, our monomers over here, our monosaccharides, there's all these hydrogens and hydroxyl groups, and they're all over the place, right? But if you come up over here, they've just disappeared, and that's actually for simplicity. It makes things easier to follow. So every time you have, I'm just going to draw a molecule of glucose. Every time you see this hexagon, we're just going to assume that's a glucose molecule for simplicity. Every corner is a carbon atom. That's a carbon, that's a carbon, that's a carbon, and we just do this, as convention so that we don't clog up the image with all these letters and stuff, right? Every corner is a carbon atom, and you can assume that every carbon atom is fully saturated, which means that carbon atom has all 4 of its bonds occupied with hydrogens or hydroxyl groups or something else. It just makes the image a lot easier to view, right? So not every hydrogen, not every hydroxyl group is shown in every corner is a carbon atom. So 1, 2, 3, 4, 5, and then the 6th one. 1, 2, 3, 4, 5, 6 carbons. So still a 6-carbon structure, it's still fructose, but we're only showing you select atoms here. All right, so this is how we build disaccharides if we're combining 2 sugars, 2 monosaccharides, or this is how we build polysaccharides. You could, in theory, add 3, 4, 5, 6 of these monosaccharides on here because poly means many. And I'll show you some polysaccharides here in a second. But how do you break these? How do you destroy these things? So let's go ahead down here, take a look. Breaking polysaccharides or disaccharides is called hydrolysis. So hydro means water. Lysis means to break or kill, depending on the context. In this case, it's to break. So we're going to break either a poly or a disaccharide in half or in pieces using water. So let's take a look at lactose. Again, lactose is a disaccharide. Here it is down here when we cordon these two off. It's a galactose and a glucose bound together. Here is the link between those two monosaccharides, and actually this bond right here is the glycosidic linkage that we're going to end up breaking in particular. Right? So we are going to add, it's the exact opposite of the other reaction. We're going to add H2O. Right? We're adding water to kill this molecule, to break this molecule. Right? This is an enzyme-facilitated reaction. In this case, the enzyme's name is lactase, which is super, super convenient because this sugar is called lactose. So we're basically just being kind of boring here with the naming convention, but it keeps things simple, right? Lactose, the sugar, needs lactase to break it, the enzyme. Anytime you see a word end in ase, it's probably an enzyme, not always, but likely. If you see a word end in ose, not always, but it's likely a sugar. So sugar, if it ends in ose, ase is usually an enzyme. I'm just going to abbreviate that, enzyme. Right. So if you look over here, here is the added water. So here is our H2O molecule. One of the hydrogens goes to galactose. The hydroxyl group goes to glucose. And we have split this disaccharide into two pieces, into two monosaccharides, glucose and galactose. So these are now two separate monosaccharides. I'll abbreviate that, two monosaccharides. All right, if you want to pause the video, take a quick little break and just try to re-explain making polysaccharides with dehydration synthesis and breaking them with hydrolysis, that would be a good opportunity. Meanwhile, let's actually go ahead and continue. So you can make molecules bigger than just disaccharides. You just simply add more and more and more and more monomers, more and more glucoses, perhaps. So let's talk about how to make these big polysaccharides, or where they occur. Oh, really quick, I'm going to go back up here. If you want to break if you want to rejoin your glucose and your galactose, so let's say you actually want to run this equation backwards, you cannot use lactase. You would need a different enzyme other than lactase. So I'll say not equal to lactase, right? Lactase and any enzyme, not only is it specific to those substrates or those products, but it's also specific to the direction of the reaction. So if you want to go the other way, if you want to say link them together and do dehydration synthesis, you would need an entirely different enzyme. All right, so back down here. Let's start with plants. So you're going to find polysaccharides in both plants and animals. One of the main ones that plants make is called starch, mostly for storage. So plants will form starch for longer term storage. That is going to be stored in a vacuole. If you guys remember in freshman year, you looked under the slides to look at a potato cell, a stained potato cell with iodine, and you guys saw the specialized vacuole filled with all the little kind of black blobs, which was the stained carbohydrates inside the potato cells, right? If you don't, that's fine, but that's where it's stored in plants. If you notice, this is a highly branched pattern. You got a branch there, a branch there, a branch there, and keep in mind, remember, each of these little hexagons is one glucose molecule. Each of these hexagons is one glucose. Right? So you have a branched pattern of glucose molecules used for storage in plants, typically stored in the vacuole. Right? And again, remember that the plant makes the glucose via photosynthesis. Via photosynthesis, right? Another thing plants can do with glucose, rather than kind of branch them like this, and again, enzymes are responsible for how those branches and how those bonds are formed. Different enzymes will actually reconstruct this glucose in a different kind of format here called cellulose. So it's not a branched pattern. I'm going to call this kind of a cross-linked pattern. And again, different enzymes will arrange these glucoses differently depending on where you are in the plant or what kind of cell you're in. You'll have different concentrations of enzymes, and that determines if you kind of make starches or celluloses. But again, these are just, again, they're all glucoses. There's nothing different about the monomer. It's all about how they're just attached to each other. All right, so these glucoses are very, very tightly knit. It's difficult to kind of break this and get materials through it. This is typical of a cell wall, right? So plant cell walls are made of cellulose. These are structural in nature. And actually, when you eat this, they're so well linked together that your body can't actually break them down. It can't break down cellulose because it doesn't have the right enzyme to break down cellulose. Lactase can break down, lactose sugar. Other enzymes can break down, let's see, sucrose, table sugar. But if you're consuming these large glucose chains of cellulose and they're linked like this, we don't actually have this enzyme to break this down. So your body passes this all the way through your digestive system. You poop out cellulose, and this is what we call fiber. So if you ever look on the back of a package and it has carbohydrates, and you look at the percent of carbs in your piece of bread or whatever, you might notice that it'll say fiber. It'll have a section for fiber, and that's just talking about the glucoses linked together as cellulose that you cannot adequately digest, that you poop out, and we call that fiber. This is very, very good at absorbing water. Absorbs water. So if you have a lot of fiber in your diet, your feces will be able to absorb much, much, much more water. It'll make them softer and easier to pass. And this is why people might take fiber or fiber supplements if they want to go to the bathroom easier. There's also other health benefits to fiber. All right, we're going to go ahead and we're going to go down to how animals store glucose. So animals will store glucose in glycogen. This is short term, and that's key. This is short term. And this is usually stored for us in the liver, in liver cells, and muscle cells. You'll hear us refer to liver glycogen all the time. So in animals, glucose is a short-term storage in liver and muscle. And if you look at the overall structure, it's similar to starch, but there's more branching. There's much, much, much more branching. You got these branches all over the place, much, much, much more frequent. And that's actually enough to make this a little bit different than starch. So more branches, I'm going to underline more branches. But again, it's just a bunch of glucoses linked together. All of these little hexagons are just glucoses. Every time you see a hexagon, we're talking about glucose, just for simplicity here, a 6-carbon sugar, right? In liver and muscle. So let's go ahead and talk about, or let's do a little application here. Let's start with plants, and you guys can write this too. We'll do plants and animals, and we'll see how these things are related. Plants and animals. Let's say that plants are going to make starch, and they do that via photosynthesis, right? They make glucose via photosynthesis, and they link all those glucoses together in the form of starch. As an animal, let's say we're doing a potato here, but potato has a lot of starch. As an animal, I'm going to eat that potato, right? And I'm going to break that down into glucose molecules. And I do that in my cells using hydrolysis, right? Using hydrolysis. I'm going to abbreviate that, right? Well, then I need to take those glucose molecules and using dehydration synthesis, I'm going to put those glucoses together in the form of glycogen. And that's going to be stored in my liver and my muscle cells, right? Well, then when I need that energy back, when I'm like running or something, I'm going to access this glycogen and I'm going to burn it. I'm going to break apart those glucoses using hydrolysis again. Ugh, my handwriting is terrible. Hydrolysis again, which is going to free the individual glucoses, which I can then use for cell respiration, right? And we're going to practice all these application things as the year goes on, but you should start to see how all of these concepts kind of work together here, right? Last little bit here over here on the side, the functions. Again, so for plants, you can have long-term storage for starch. For animals and plants, you're going to have to use this for cell respiration. Animals are not the only things that do cell respiration. This is going to happen in the mitochondria in both plants and animals. If you have cellulose, obviously that's going to be structure. Again, this is just many, many glucoses. I'll say many glucoses. Another thing that is a structural, is structural besides cellulose that we'll talk about throughout the year is called a glycoprotein. Glyco means sugar, protein means protein. These are going to stick onto the cell membrane and they're going to be used for signaling. So we'll talk about proteins with sugars on them, with glycogen groups on them, and we'll get there next unit. And then obviously for storage. So again, starch is in plants, that's stored in vacuoles. Glycogen is in animals. I'll abbreviate this. Animals, and that's going to be in liver and muscle. I'll abbreviate that. And remember that glycogen in particular is short term. Short term. All right, so that's it. Hope this helped, and I will see you guys next time.